All right. Let me present Dr. Chelsea Specht. She is the Barbara McClintock Professor of Plant Biology and Associate Dean for Faculty at Cornell University. She is also a member of the LH Bailey Hortorium and is leading a new biodiversity initiative at Cornell University that are driving innovations in basic and applied biodiversity research. Chelsea received her PhD in biology from the New York University as part of a joint program with New York Botanical Garden. Her research group uses traditional morphological and developmental techniques combined with molecular genetics, population genetics, comparative genomics, and phylogenetics to study the natural diversity of plants and to help understand the forces creating and sustaining biodiversity. Go ahead. That's it.
Awesome. Thank you. Great. Well, I personally, my first for today is I've never worn a headset like this before, and I feel a little bit like a cross between Taylor Swift and Madonna. So I'm not sure which I'm going to channel at the moment, but anyway, I do not have the energy of Taylor Swift. Boy, I don't know if how many of you have gotten to go see her Eras tour? Anyone here? A couple in the audience? All right. I have a 14-year-old daughter, so I did get to go, and man, I have never seen somebody traverse a football field so fast in high heels. So today, in honor of being Costa Rica, and I'm going to change the title of my talk a little bit, in honor of being in Costa Rica and literally surrounded by zingiberales, I'm going to center my talk on our latest research on Costaceae with a focus on New World Costus. I want to be sure to touch on some of the practical applications of how our work is informing best practices for biodiversity research, using genomics as tools to build biodiversity literacy for students, scientists, and communities. And I'd love to engage in conversations with you throughout this meeting, this conference, on ways that we as a community can co-create research that most directly addresses the biodiversity crisis through gathering and creative deployment of biodiversity knowledge. In addition to my co-authors for this talk, I'd like to thank Team Costus, which is Eugenio Valderrama, Paulin Hilchi-Moss, Thiago Andre, and Dave Skinner, for creating an environment in which we can together share, explore, and distribute our passion and expertise for Costaceae as a collaborative venture. I'd also sincerely like to thank the organizing committee and all of the organizers as individuals, in particular Melania Fernandez and for her support in getting me here today, and Adam Karamans for all he's been doing to make us feel at home in this beautiful country. So the genus Costus comprises at least 106 species with a distribution across the tropical forests of Americas and Africa. The neotropical radiation is fueled by the diversification of organismal interactions, with guilds of pollinators often associated with particular species. Both hummingbird attraction and more recently bee avoidance are considered to be possible drivers of the coloration of flowers and inflorescences. The red bracts and the red to orange tubular flowers associated with hummingbird pollination may be involved in attracting birds and may involve the avoidance of bees, a hypothesis that posits that dark-colored inflorescences and flowers may have evolved to discourage visitation by nectar-robbing insects. In addition, the presence of ants is well documented as a strategy against herbivory in many plant species, and the extrafloral nectaries and the indiment of Costus are both traits that seem to attract ants to live on the plants, thereby reducing herbivory by larval predators. The presence of ants could thus also impact pollination biology, as bees tend to avoid flowers that are covered by ants. So given the number of evolutionary events in which bird pollination evolved from bee pollination throughout the Neotropical Costus, they provide an interesting case for further investigation into the evolutionary drivers of trait evolution, be they the attraction of effective specialist pollinators or the avoidance of less effective generalist pollinators. We hypothesize that the evolution of Costus inflorescence morphology reflects an interplay between selective pressures related to pollination biology and herbivory, influencing trait evolution and the tempo and mode of diversification across this tropical lineage. So while as a team, our relationship with Costus goes back 60 years, including that with Paul and Hilchi Maas and Paul's early dissertation work on Costus. This specific project that I'll be talking about today started just over a decade ago with the desire to monograph the New World Costaceae, including the Neotropical Costus. The phylogenetic context that emerged from this revisionary work has demonstrated that morphological innovations associated with pollination efficacy are highly homoplasious and appear in combinations that impact pollination behavior. The goal of this research is thus to further investigate evolution and diversification of whole genomes and of candidate genes known to be involved in the evolution of morphological traits, and to look for signatures of selection that may be responsible for driving the rapid radiation in this lineage. So in order to get at this question of convergent evolution, we first need to build an updated phylogenetic hypothesis for Costus, and then investigate morphological changes associated with pollination and identify signatures of selection across loci and comparing whole genome-wide signatures. So really this starts with building the phylogenetic hypothesis. And at this point, I'm sure many of you in the audience are like, don't you already have one? Haven't you been doing this since, I don't know, 2000? So what I'd like to, I think, and Tom Givnish gave a really good introduction to this, and the progressive nature of phylogenetics and systematics, in that it's a progressive effort that takes additional sampling, additional data collection and data analyses, and it moves towards recovering resolution, especially in rapid radiations. So the acronym for recovering resolutions for rapid radiation is ER, and I think that's oftentimes how we feel when we're trying to get at those difficult phylogenetic questions. So what I'm going to talk today talks about a captured loci, so a HYBE-seq approach, and comparing this with whole genome studies, where we can use SNP data, or single nucleotide polymorphisms, to provide a different level of resolution that might allow us to get at different types of questions. What can we do with these different data types is something that I hope to address today. And then also a question that I found particularly interesting recently is how to add a new species to an existing massive data set. So in the past, you get something new, somebody brings it in from the field, and you're like, oh, let's get the RBCL and some 18S and sequence them out, and then we can tree it, right? We can place it in the tree and we'll be fine. But now with these massive data sets, you've got this new taxa coming in, and you kind of wonder, what do I do with it? I don't have a full plate for sequencing. And so I want to address that today. How do you place a new taxon into an existing massive data set in phylogeny? So our baseline data that we're going to start with is a HYBE-seq capture approach that we published, Eugenio Valderrama, Jacob Landis et al. Published in 2022. This used probes that were designed for Costus, from Costus. The loci were identified as single copy and informative for phylogenetic analysis. It has 240 terminals and over 1 million base pairs of data. And we used concatenation as well as coalescence-based trees. And one of the things that this allowed us to do was to place 17 new species that we described in PhytoKeys in 2023. This allowed us to test reciprocal monophyly of these new species, potential new species, and then incorporate this evolutionary phylogenetic information into the monograph, which is currently under review in Blumea. We also used this phylogeny to identify shifts in bee and bird pollination, and this allowed us to select pairs that we then decided we were going to use for whole genome analysis to test for those signatures of selection that I talked about in the introduction across these different pairs. So we've got bird pollination, bee pollination, closely related taxa. Where might we see different whole genome level signatures of selection? To get at those genome-wide evolutionary trends, at this time, we used Illumina coverage for, again, 26 species that were developed into these species paired. And we called SNPs, or single nucleotide polymorphisms, from the genomic data. At this point, we took a little bit of a pause because we realized that the SNP data, in addition to giving us information about evolutionary trends and selection, signatures of selection, could also be used for phylogenetic analysis. So why would we pause and think about using our SNP data for phylogenetic analysis? After all, we only have 26 taxa that we have this SNP data for, and our other phylogeny has 244 terminals. Well, for one, SNP data is easier to generate. You can sequence whole genomes, and I'm going to say easier. I'm going to put that in a little quotes. We can talk about that after the talk. But you can use various different methods. You can use genome skimming or RADseq, genome resequencing. Many of these are used for population genetic studies and then can be co-opted to use for phylogenetic study. And if you've taken all this time to generate one data set, you might as well use it for as many things as possible. Also, SNP data are distributed across the genome, so that means they represent a number of unlinked loci. And this paper that I'm highlighting is by the Jacob Suissa and Jacob Landis, is in review right now. There's a bioRxiv preprint. You can ask me for it later. And it's about guidelines then for using SNP analysis, for using SNP data in a phylogenetic analysis. And without going into any of the details, I want to give you two take-home messages from this. One is filtering. The first thing you have to do when you get SNP data is to filter it across a bunch of different parameters. Just want to say that over-filtering can lead to vastly inaccurate branch lengths, significantly inaccurate branch lengths, whereas under-filtering just allows you to spend a lot more time doing computational analysis without getting any better resolution. So filtering is important. And then also, if you're going to do downstream phylogenetic comparative methods, you want to have a chronogram. This allows you to remove some of the branch length bias that occurs in phylograms when you're comparing SNP versus locus data. But in our case, we use both concatenation and coalescence to look at our SNP data. And we have the concatenation tree here, the coalescence-based SDVD quartets tree here. And you can see that with our taxa, they're largely congruent. And these are also congruent with our larger phylogenetic analysis of the 244 tips. There's one place where they are not congruent, and this leads to a poorly supported node in the SVD quartets tree. And this corresponds to the position of Costus venosus, which has highly variable colors, and we do have a sequence that we know is a known hybrid. So there's a high potential for venosus to be a hybrid, which brings in another really great use of SNP data, is that you can, using splits tree, you can create a phylogenetic network. And this network, rather than assuming a bifurcating topology, allows you to see where you have incongruence in your sampling that is the result of multiple genotypes contributing to the genotype of an individual. In this case, we can actually identify that Costus venosus is part of one of these network parallelograms, as well as the species mellow yellow, which is a known horticultural hybrid. Love having horticultural hybrids and being able to throw them in the analysis to test things. So we can identify the likely maternal progenitor, Costus phylicissimus, of our Costus venosus hybrid, as well as potential other progenitors, Costus wilsonii and Costus pictus, both of which are found here in Costa Rica, actually all of which are here in Costa Rica, which may be involved. So SNP data does do a really good job in allowing you to identify possible parents of hybrids, and it does this better than many of the other data sets that I'll talk about subsequently. Also, this allowed us to place a new species. So we sequenced the genome of something that we thought might be a new species, and included that in this analysis. So we have our SNP data for that particular species. And we figured, so it sat here with the SNP tree, but because we were testing this, we also used HiveSeq data for that same, we have 3 accessions of that individual. And so we used HiveSeq data to ensure that, yes, that individual is monophyletic. The 3 taxa come out together, they're reciprocally monophyletic, and then to place it into our larger phylogeny. So we had 3 samples, we placed this into our larger phylogeny, and we found that although this has more taxa in the HiveSeq phylogeny, the Costus new species placed in the same clade in the same in both phylogenies, indicating that our SNP data would have given us at least relative information about where this new species belonged and correct information about where it belonged. Now, how many of you know pollen Hilchi moss? Okay, so then I need you guys to be quiet for a little while. This is a secret. So this is the new species, and we're naming it after them collectively. So it has a name that is collective, and I'm not going to give out the name yet, but I just want to let you know. This is like a secret, so don't tell anybody. We'll hide this from them. They don't know, right? They don't know. And it's been really hard to hide a new species from like the 2 amazing taxonomists that they are. So I hope I didn't just give something away that I didn't mean to, but it's a beautiful new taxon. Oh, and one of the things that's special about it is you get 2 flowers at a time. And in most species of Costus, only 1 comes out at a time. Okay, so I just talked about our SNP-based phylogenetic analysis. And as Tom Givnish alluded to yesterday in his talk, we also have a bunch of other taxa that are a bunch of other markers that are coming out that can be useful for phylogenetic analyses. These are ultra-conserved elements, the angiosperm 353, as well as the Busco genes. And we also have our HYBE-seq data set. And at some point, we realized that with our genome resequencing, we could use that genomic information and bioinformatically capture the ultra-conserved elements and our HYBE-seq loci from those genomes. So we already have HYBE-seq from those 26 taxa, and now we can go ahead and see how our bioinformatically captured data might compare to our actual sequenced and captured data. So I want to go into this just a little bit to describe how this can work, because you've got your genome skimming or your 10x covered genome sequencing, and you want to extract, again, as much data as you can from that. So what we did is we took our bioinformatically captured the angiosperm 353, the Busco genes, which are these purported universal single copy orthologs, and our Costus capture probes from those genomic data sets. We then investigated what kind of recovery we got using this bioinformatic approach. Was there more porology? What was the porology looking like? Because with the angiosperm 353 and the Busco genes, we're not sure what the porology might look like in our particular taxa. And we looked for support. Where are we going to get support to recover these topologies? We're going to compare this with our true tree, which is our SNP-based tree, which we know reflects the phylogenetic relationships of our larger capture data set. And then talk about where we can use this to add new taxa to phylogenies. So we wanted to figure out where we might stand in this. And this is the monocot tree of life. And we took all the genomes that occur for monocots that are published. And we used and this is work that Josh Felton, who's in the audience somewhere, has been working on together with Jacob Landis. Can't see him, he's somewhere out there. And they captured the angiosperm 353 targets from these monocot genomes. I just want to point out a couple that seem to be, and the percentages here show the percentages of targets that bioinformatically mapped to angiosperm 353. So you would expect to find all of them might have the angiosperm 353 targets in them, considering that these are conserved genes. But you can see the mapping, at least at the whole scale across the genome, showed that it was slightly less. And in fact, there was a low percentage of targets mapping to the genomes in several of the species highlighted in orange. We also felt that we wanted to look for multiple mappings, and so that's the second number shown here. And you can see that in the grasses and also in asparagus, there's a high potential for multiple mappings and porology of these taxa. So these are two things that you're going to want to look for when you're doing this for your own taxa. Within Costus specifically, we also saw the same sort of trends, and we had an expectation that there was going to be low recovery and moderate levels of porology for the angiosperm 353 data set. What we found was actually quite different. When we went in and used the sort of more rigorous protocols to look at the maps, we found that all 353 loci did have reads that mapped. And in fact, 343 of those assembled to 150% of the target length. So that means we're assembled not just the angiosperm 353 loci, but also flanking regions or potentially intronic regions that could give us additional information for phylogenetic analysis. What this graph shows is that everything in this sort of pale color is something that captured and was only found in a single copy. So these are our non-porologous genes. Those in the darker colors do have porology warnings. So these are things you'd want to look at and make sure you identify the appropriate ortholog. And in Costus, 7.4% of had porology warnings. And then we did have some taxa that didn't capture very well at all, and that might have something to say about our genomic coverage. In this case, it was for something that we used an herbarium herbarium sample for. So recovery across the target sets, I just showed you what we did for angiosperm 353. We did the same thing for the Busco loci as well as for our Costus capture loci. And we found that 95% and 95% respectively, I forgot those numbers were the same in my head, for angiosperm 353 and the Busco loci. So we're capturing almost all of those loci in our genomic sampling. For the Costus capture, it was slightly less at 87%. Our porology is the highest in the angiosperm 353, less in the Busco, and it's 0 in Costus, which makes sense because those are the captures that we designed to not give us porologous loci. And we have quite a few points of data. We have quite a few base pairs. I just want to point out that the SNP data set is 5.5 million base pairs, and it's unlinked loci. So this is comparable to what you might see in the BUSCO loci, but as far as numbers are concerned, but they are unlinked as opposed to the BUSCOs, which are linked loci as they're part of single genes. So how does the resolution compare? And I'm going to go through this a little bit quickly, but I just want to get to the point that, as you know, we're doing this for a rapid radiation. We wouldn't expect that the resolution was great for the angiosperm 353 or BUSCO. Just want to point this out, but our anticipation was not that the resolution would be fantastic. If you're doing this for a different level of phylogeny, though, a different level of analysis, a more deep time, you would expect your resolution to be really good. So yeah, as expected, the angiosperm 353 topology was not very congruent with our SNP phylogeny. You can see that by all the lines here crossing over. This is a way that Jacob and Josh looked at these trees at this analysis. What you see here is in blue. This is a particular node that's supported by all of the loci in the analysis. All 345 loci supported this node. However, when you look throughout the rest of the tree, the pink color indicates that there's conflict. So the individual trees, the individual gene trees from these loci are conflicting with the overall topology at almost every single node. So I want to introduce another measure that they're using to look at this. What you see here is a graph that shows all of the dots are different loci, and along the axis here, you see numbers of nodes that those loci support that as a gene tree that are in the final overall phylogeny or overall species tree. There are 24 total internal nodes, and for the for the angiosperm 353 data set, only 4 nodes were like a locus. The average locus at 50% only supported 4 nodes, meaning that just each loci just doesn't have enough information to support nodes in the phylogeny, which again, fully anticipated given that this is a highly conserved data set and we're looking at a rapid radiation. The same is true for the BUSCO genes. Again, highly conserved data set, we're looking at a rapid radiation. Somewhat better support, but still the median locus does not support very many nodes in this analysis. And you can see that there's not a lot of congruence between the 353 and the BUSCO genes. So then we want to say, how does our Costus-specific targets do in this type of analysis? They do better. Still, there's not a lot of support per locus, even though these were loci that we particularly chose because they would have phylogenetic information. However, the overall phylogeny is quite congruent with our SNP topology, with a few exceptions, and those exceptions are taxa that are jumping around. Those are our hybrid taxa. So what we have here is that we've got loci where each individual locus does not provide high support for our topology. However, the combination of those loci together does provide congruence with our expected topology that we got from SNP data. So our goal for doing this was not to see if we could get a resolution from angiosperm 353, because we knew we couldn't. But what we really wanted to do to see if we could identify loci that we could cherry pick. And those loci would give us an idea of what the phylogeny might look like. This is the idea of barcoding, right? You choose a couple of loci that might give you the same resolution that you would expect if you had the whole tree. And what we kept thinking is we have a huge HiBSeq data set. What if we find a few genes that might work that would give us the right resolution? And then we can sequence those genes out of a few taxa, add those to the phylogeny, and place our taxa into a phylogeny. But what we find is that even if we cherry pick these loci, they don't really provide accurate resolution of where those individuals might go on the phylogeny. So we were kind of unsuccessful in our ability to find barcoding loci from this analysis. No single locus or few loci were able to resolve the nodes or to provide correct location of individuals within a resolved topology. So as we know now, the power comes from hundreds of loci and deep taxonomic sampling. And if you want to add a new taxon to a data set, the best result would come from doing some kind of genome resequencing, getting about 10x coverage, assembling the loci with HiPiper of your whatever loci that you're using for your larger analysis, and then calling SNPs bioinformatically, calling SNPs or bioinformatically calling those loci, as we did with the Costus loci. So when you're thinking about making your commitments to data gathering, one, get as much taxonomic sampling as you can. HiveSeq, targeted to your taxon clade, will give you not only data for that particular data set, but in perpetuity as you do genomic level sampling. You can use this genome resequencing approach not only to get SNPs, but then also to bioinformatically capture loci, taxon-specific loci, as well as ultra-conserved elements that you might be using for other analyses. Again, the SNPs approach is going to give you great information about rapid radiations where the angiosperm 353 and BUSCO genes are going to work best in deep time. Okay, so now I'm going to back away from all the phylogenetics and go back to what we originally got this SNP data for, which was to get genotype calls for looking for character trait association and selection analysis. And I'll say also from the genome capture, we could use this to get candidate genes that we might think are involved in some of the traits that we're interested in observing. So now this is moving into the second part where we've got our phylogeny. We're going to investigate morphological changes associated with pollination syndromes and identify signatures of selection. So now we get to talk about the traits. We're moving from the trees to the traits. What we did for Costus is we scored features of the inflorescences that could be related to pollination syndromes as binary traits for the included taxa. The traits corresponding to pollination syndrome are labellum shape, which directly corresponds to observations of flower visitors, with hummingbirds visiting flowers with tubular labellums on the top and bees visiting flowers with the spreading labellums. We also looked at bract color, bract appendage, present or absence, a nectar guide or the honey mark, which is present in the top, which is a yellow spot in the center of the flower. Labellum stripes that also seem to play a role as a nectar guide down to that center honey mark. And then finally, functional color. And I'll describe functional color a little bit later, but this is the idea that a taxon can have red bracts, but because the appendages might be a different color, those appendages give the color of the inflorescence. So bract color doesn't fully give the color of the inflorescence. The color of the functional color of the inflorescence is derived from the bract color, the bract appendage, and the bract appendage color. Presence or absence of bract appendage, and then the bract appendage color. We also scored traits associated with herbivory and possibly pollination as well. These are inflorescence indiment and the presence or absence of an extrafloral nectary, shown here as this callus that submits nectar. Using stochastic character mapping, we investigated the evolution of each of these scored floral and inflorescence traits. All traits were mapped onto our existing phylogeny, and then we did a Pagel's analysis of correlated evolution to see what kind of correlated evolution you had among these traits. What we found is that all traits do have phylogenetic signal, and that correlation exists among certain specific traits. Labellum shape, functional color, honey mark, and labellum stripes were all highly correlated, and bract color shows a weak association with those above traits. The callus and indiment were correlated with the presence of bract appendages, which gave us some new insight that we hadn't really thought about before, is what is the role play of having this appendage and how does that interact with the presence of the extrafloral nectary. The labellum shape is the trait that is highly correlated with what we had previously coded as pollination syndrome. The tubular labellum is likely to be ancestral, and other characters related to pollination, to any pollination shift, occur after changes in labellum shape, meaning that trait shifts follow these expectations for the pollination syndrome. Once you develop a spreading labellum from a tubular labellum, you then see those taxa develop green inflorescences, stripes on the labellum, and a honey mark. Bract appendages evolved multiple times independently, and only 1 to 2 times has this led to a clade of appendage species. One of those times is a clade of species that is incredibly prevalent here in Costa Rica. And in fact, I was very excited in Diego's talk yesterday, where 2 of these species, Costus montanus and Costus barbatus, were mentioned as taxa that are under concern for, they're in national parks, so they're not in concern for conservation measures. But all of these taxa, but my favorite being named Costus ricus, named by Paul and Hilchi Maas, super creative. As well as Costus L, which is from the Osa Peninsula. The callus in the bracts has been lost in several lineages independently, while inflorescence indument has been gained independently in several instances. And you can see situations where you have the loss of the callus in the bracts being lost and indument being gained and expressing throughout these clades. So this is an upset analysis, and it's an analysis that allows us to code these different characters where we're looking at appendages, labellum stripes, the same characters here. And if there's a dot, that character is present. So this is a functionally red, tubular labellum, and the bracts being red. In this case, you have labellum stripes and a honey mark with absences here. So that means it's a green, functionally green. The labellum is spreading, not tubular, and the bracts are green. What this allows us to do is look at the taxa, the number of individual species that contain this suite of traits. And the most prevalent combinations of the analyzed traits match our expectations for pollination syndromes. The most frequent combination corresponds to the morphology associated with hummingbird pollination, with a tubular labellum, overall red inflorescences, and the bracts being red. The second most observed combination agrees with bee-pollinated inflorescences, with a spreading labellum, with stripes in the honey mark, and green inflorescences, and no appendages on the bracts. The third and sixth most common combinations correspond to the same hummingbird and bee pollination morphologies, but with appendaged bracts. The fifth most common indicates red bracts, so this would seem to be in conflict with the previous morphologies. However, this species Costus leucanthus, while it does have red bracts, also has green appendages that conceal the red bracts, making the functional color of the inflorescence green, not red. This means that even with red bracts, Costus leucanthus has the same pollination morphology as Costus guanaiensis, making the fifth and sixth most common morphologies functionally identical to one another. You can see the same trait of red bracts concealed with the green appendages in this individual, Costus splendii. So you can see that those red bracts are in there. And so when you're coding this for a monograph, you're coding it as bracts red, but the functional morphology is green. The fourth most common combination is functionally identical to combination 2, but without appendages. So what's interesting from this data set is that it shows that it seems easier to mask red bracts with green appendages than to get rid of the red bracts altogether or to turn them green. Other observed combinations shown here, this tail, do not match the expectations of any pollination syndrome and are less frequent. And so we wanted to see what kind of transition rates occurred and whether or not these individuals might be species that are sort of caught in transition to one of the pollination syndromes. And indeed, that's what our data show us. So what I'm showing here are transition rates for correlated evolution that indicate how pairs of traits evolved in concert. We're looking at labellum shape and functional color, and the strength of the arrow indicates transitions toward those combinations of traits. The most stable combination of characters match pollination syndromes, shown here as tubular and red, or spreading and non-red. And note that the non-red functional color can be obtained either by changing to a green back color or by gaining leafy appendages. That's important to note as you're looking at these. The alternative combinations, the tubular non-red and the spreading red, are very unstable and quickly transition to the more stable combinations. In the model of correlated evolution of the labellum shape and the bract appendages, the transition rates of the appendages are higher in lineages with a spreading labellum, suggesting that appendages are more labile in the lineages with the spreading labellum, which is also associated with bee pollination. Similarly, when appendages are present, so here we're looking at callus and appendage or indument and appendage, when appendages are present, the transition rates for gaining or losing the callus in the bracts are higher. And interestingly, the lineages with indument on the inflorescence show even higher transition rates for the presence or absence of bract appendages. This is indicating that indument and bract appendage presence is somehow correlated. This is something we're trying to contemplate, but it might have to do with attraction of ants and herbivory. Hairy inflorescences provide some pretty excellent living conditions, which may be responsible for deterring some forms of pollination that involve larval deposition. I should point out that we found no evidence of differential diversification rates related to labellum shape, so consequently to pollination syndrome. Okay, boy, I have a different time. So looking at signatures of selection real quickly, I just see I have 3 minutes and my time is off on my timer, so I apologize for that. I'm gonna go a little bit more quickly. So going back to these genome resequencing sampling, we optimized high throughput long sequencing reads in the process of this. I just want to point out that this is published in Applications of Plant Science by Giselle de la Cerda, and great methods, happy to talk about this later, about how we got from genomes to understanding some of these patterns of evolution. We found 44 candidate genes that we investigated for signatures of selection that were involved in symmetry, color, flower size, and boundary formation. 11 of these showed evidence of positive selection. We see elevated selection. This shows a branch-specific test for selection of those candidate genes. And we found elevated selection on species with tubular labellum versus non-tubular labellum. We also saw this is in petal loss gene, which is the gene that has to do with fusion and floral formation. We also saw this in Superman, another gene involved in floral formation, and actually in the potentially involved in the infertility of the labellum in Costus. It's not as significant, but we did see the same sort of trends in Superman. We then looked at 2 genes that were involved in color formation, functionally red pattern. They're both chalcone synthase genes. In one of them, we found no difference in selection between functionally red and not functionally red, indicating that the chalcone synthase locus CHS4 may not be involved. However, another locus of the chalcone synthase gene, we found that species that are not functionally red show elevated values indicating positive selection. And so finally, I just quickly want to mention linkage disequilibrium analysis, because we found that there was a higher degree of decay, of linkage decay in hummingbird pollination versus bee pollination. And outcrossing and hybridization can lead to a rapid decay of linkage disequilibrium, making us wonder if hummingbirds may increase the possibility of outcrossing among genetically distinct individuals, leading to this faster decay in linkage disequilibrium. So that's something that we want to consider. Then we were also able to use the data to look at selective sweeps, and we have future directions in this to look at possible genes that are involved in some of these selected sweeps, and also we need to get population level sampling to further investigate this. So in the interest of time, I'm not going to summarize, except to say that we have a lot of genomic data. We were able to do a lot of fun things with it that allowed us to understand some of the patterns and processes of diversification of this amazing group of Costus. So I'm going to take my last minute to introduce something that we've been working on in the last couple of weeks, which is a key to the New World Costus as an interactive key. I will give the website to this at the end. I do have to ask, though, that not everybody go on it at the same time because it will crash the server. And I've been asked not to give the QR code, but of course I'm doing it, because just go ahead and take the QR code, but then use it later, because all of you are going to go out into the field after this conference. You're going to see Costus, and now you're going to be able to identify those Costus. We have a key specifically to images of Costus of Costa Rica here, but you can also use the key to get down. So I just want to show you quickly how this works. This is an algorithm that was developed by Kevin Nixon at Cornell University. He's been wanting to test this for a long time, and we're testing it now. So what you'll do is you'll get this, when you go to the QR code, you'll get this page, information about the genus Costus, and then you can click on New World Costus Key. This will take you to a page that allows you on your phone to see all the taxa that start at the beginning, and then select characters that will help you distinguish among those taxa. If you don't have the character available to you, just hit next, and then you can go to the next available character. But in this example, let's say we have this all 76 taxa, and we select that it has a yellow corolla color, then hit next. The next thing that comes up is sheath indiment. Of course, you're looking at an herbarium specimen or a taxon in the wild, and you can see that it has a brownish indiment. Again, hit next. It will give you another set of characters. You're now down to only 9 possibilities. In this case, you're looking at bract appendage color, and the specimen that you're holding has green appendages on its bracts. If it doesn't have appendages, you can click this one here. You can also click combinations of traits. So if it has two or it's ambiguous, you can select both. That's fine. We love ambiguity. And then finally, the last one is calyx length. You've got only 5 taxa that it can be, where it has greater than 20 millimeter calyx. Again, you hit next. 2 species possible that are left. Divide it between whether it has the honey mark. Yes, it's present. And then you can get to your 1 taxa identified Costus longibracteolatus. The way this data set works is we have a species page that has the description that's from our monograph that will hopefully be published soon. You can also connect to Dave Skinner's Gingers Are Us database, which has a bunch of information about the species, including all of his observations, and he's an amazing observer, so he has fantastic observations. And from this, you can look at his field observations as well as his Google page. So here's the QR code. I'm gonna leave that up for a few seconds. Again, don't use it right now because Kevin is sitting at his computer at Cornell freaking out, afraid that everybody's gonna start using it immediately. So don't use it immediately. But do let him know that show him some love. And with that, I would go to my thank you slide, but I want everybody to get an opportunity to use the QR code. So I'll leave this up for a little bit, and then I'll put my acknowledgement slide back up. Thank you all.
Thank you so much, Chelsea, for that. We won't have time for coffee, so no.
I swear it only says 39 minutes on mine.
That's fine.
I must have done it wrong.
A quick question.
Yeah, Regina.
Thank you, Chelsea. Thank you very much for this interesting talk. So I'm interested in the shift from bee to bird pollination, and I have the specific question: do you also find a reversal to bee pollination?
Within the taxa, do we see reversal to bee pollination? So yes. However, it's sort of single characters. So we do not see at this point where you get a full reversal to bee pollination where the full suite of characters comes in. So it seems to be that there's still this transition. So you go from bird pollination to bee pollination. So you do not see that transition back to the fully tubular labellum. That seems to be hard to get. So once you go from bird to bee, you now have the open spreading labellum. To go back to full bird pollination syndrome, you'd have to go back to that tubular labellum. And that seems to be the aspect that doesn't happen yet. It could be because the combinations of traits isn't there, so the selection pressure isn't strong enough to go back to a tubular labellum. Or it could be that you can just change your color and you don't really need to have that ultra tubular labellum in order to effectively be pollinated by hummingbirds. So it is an indication as to whether or not the selection pressure is acting, like if you have all the other suites of characters, do you need to get that final character that would pull you to the tubular labellum, or can you do okay with by having other characters that attract the pollinators and repel the bees, if that's part of the process? Does that get at your question?
Very quickly, please.
Hopefully a short answer, but in the A353 analyses, did you focus on the protein coding parts, or did you also investigate the more rapidly evolving splash zones?
The splash
that might
zone
be useful for rapid release.
Yeah. So I mean, as I said, we got the 150%. So we used that entire amount. What we could do though is go back in and cut out the protein coding part and just include the splash zone. It'd be fewer loci, but that could be one way to go at it. We could also work on those bioinformatic approaches to make sure we got more of that splash zone. The other thing I was thinking about is when developing the probes, there could be ways that we could go back to that bioinformatic probe development and intentionally create some ambiguity in the probe so that they would get more of that splash zone. Yeah. And you could do that on a taxon-based approach too. Yeah.
All right, thank you so much. We'll head out for a coffee break and reconvene. You're eager for coffee, I see. All right.